Dear students, I am Dr. Sonal Mishra, posted in Government Digvijay PG College, Rajnandgaon. This is the fourth part of this series. In first part, we have dealt about the requirements, method, techniques, and types of tissue culture, plant tissue culture. In the second, we have dealt about somatic embryogenesis and androgenesis. In the third part, we have dealt about the artificial seeds. And this is the fourth part. Today, we are going to deal about the production of hybrids and somaclones. Today, these were the topics of discussion: production of hybrid, somaclone, and somaclonal variations. Production of hybrid. Hybrid seed production. In agriculture and gardening, hybrid seed is a seed produced by cross-pollinated plants. Hybrid seed production is predominant in modern agriculture and home gardening. It is one of the main contributors to the dramatic rise in agricultural output during the last half of the 20th century. Today, we can get each and every vegetable throughout the year. All this has happened because of the new techniques in which hybrid seed has contributed a lot. Hybrids are the species developed by the crosses between different species of plants. This is the cross between two genetically dissimilar individuals having difference in their genotype. This process is known as hybridization. In this process, the characters present in two or more species of plant are brought in one single plant. For example, one plant is high yielding plant but it is prone to diseases, while the other one, which is disease resistant plant but having less yield. The two good characters can be bought in one single plant by the process of hybridization. That means we can create a plant C with disease resistant capacity as well as high yielding capacity. The resulting seeds are called hybrid seeds or F1 seeds, and the plant produced is called hybrid plant. Hybrid. Hybrid is formed by combining the qualities of two different breeds, species or genera through sexual reproduction. What is hybrid plant? A plant that has been produced from two different types of plant by combining them. Hybrid breeding, when two different plants are crossed together to produce a new hybrid variety, it is called as hybrid breeding process. Advantages: in most of the cases, the hybrids are better than their parental ones. Hybrid and hybrid breeding. For example, one plant we have taken as a parent 1 is a long plant, while the plant chosen as parent 2 is a dwarf plant. And when they are crossed, we will get another plant in F1 generation, that is first filial generation. That plant would be long hybrid plant due to the dominant characters represented by the parent 1, which one is tall plant. Thus, a new plant is formed by the crossing of two different plants. This plant is known as hybrid plant and the process is known as hybrid breeding. Process of hybrid breeding. Hybrid is produced by crossing between two genetically dissimilar parents. Pollen from male parent will pollinate, fertilize and set seeds in female to produce F1 hybrid seeds or first filial hybrid seeds. For production of hybrid, crossing between two parents is important. The crossing process will result in heterosis. In self-pollinated cross, it is difficult to cross, but in cross-pollinated crops, it is quite easier. Types of hybrids. Interspecific hybrids. Hybrids between two different species of the same genus are known as interspecific hybrids. For example, Dhatura. Intergeneric hybrids. Hybrids between the plants of two different genera of the same family, like potato, tomato. Intertribal hybrids. Hybrid between plants of two different types are known as intertribal hybrids. Procedure. First important step is selection of parents. It depends upon the aims and objectives of breeding. The superior plant with all good character is used as female parent, and the plant from which characteristics have to be transferred or the character we have chosen to transfer should be selected as male parent. That means the basic plant in which we have to modify the things is chosen as female parent, while the plant from which one or two characters we want to transfer is chosen as male parent. Selfing of parents to induce homozygosity. Emasculation, removal of stamens for hermaphrodite flower before maturation of anther is known as emasculation. This is done to prevent the process of self-pollination. It is done usually in the evening and cross is made in the next morning. During emasculation, gynoecium should be intact. The anthers are removed with the help of forceps carefully. Those flower buds are selected which are going to open in the next morning, because this is to be done before the anther dehiscence. It is done with the help of hands or forceps very carefully. Flowers are collected and the cones are taken out, then they are dried at 30 degrees centigrade for 24 hours. Then put the dried anther cones in a cup, then transferred to pollen lead cup, and the process of pollination is made with the help of hands. Bagging and tagging. The emasculated flowers is immediately bagged to prevent cross-pollination by any foreign pollen, because pollen grains are found suspended in the air. After emasculation, flower buds are covered with butter paper or plastic bags and dried firmly. Tagging. The bagged flower is tagged by level crossing. In the next morning, the desired pollen is placed on the stigma of emasculated flower to bring about fertilization. Harvesting and sorting of F1 seeds is done, then raising the F1 generation. This is the process of bagging and tagging. It is covered with the help of polythene and proper tags are put on the plant. Hybridization of self-pollinated crops. First method is bulk method. The bulk method of breeding was first used by Nelson Hill in 1908. This method is also known as mass method or the population method. In this method, in the bulk method, plants of F2 with desirable traits are selected and work together to raise the next generation, that is F3 generation. Seeds from selected F2 are shown many times and the process is continued till we obtain homozygosity in the plant. In this plant species is grown in bulk plots up to F1 generation till F5 generation. That means from first filial generation to fifth filial generation with or without selection. The duration of bulking may vary from 6, 7 to 30 or even more generation up till we will reach the homozygosity stage. When desirable purposes are fulfilled, bulking is stopped and individual plants are selected, evaluated for the characters and introduced as a new variety. Outline of this process: in season 1, select most desirable parents, make educate number of artificial hybridization. In season 2, plant F1 seeds which we get in season 1 and grow under favorable condition. Harvest the bulk F2 seeds which is generated from F1 plants. In season 3, plant F2 seeds similar to commercial conditions. Harvest F3 seeds and bulk them. Repeat procedure until F6 generation, then select individual plants and harvest seeds from selections, keeping seeds separate. Season 8, plant progeny rows of each selection, select best uniform rows, harvest seeds from each selected row and bulk seeds from the row together. From season 9 to 11, test performance of selections is replicated trials with standard check cultivars. That means from 9 to 11 generation we will test the characters, its performance. Then in season 12, increase lines that are superior to check cultivars and then it is released as new cultivar. Here it is shown in the diagram. Two plants are selected as plant P1 and P2. They are the two parents. By their crossing, first filial generation is produced, that is F1 generation, which is bulk planted. This is repeated up to F2, F3 and F4. In F5, they are space planted. The selection of superior plants is done from the bulk plantation and they are space planted. In F6, they are selected and planted in different rows. In F7, primarily yield test is done from F9 to F10. Advanced yield trials is done and then it is finally released as new cultivar. Second method is pedigree method. The method was first described by Lowe in 1927. Pedigree, a description of the ancestor of an individual, and is generally goes back to some distant ancestors in the past. Thus, a pedigree would describe the parents, grandparents, great-grandparents, and so on. The pedigree is helpful in finding out if two individuals are related by their ancestry and therefore are likely to have some genes in common. In pedigree method, a detailed record of the relationships between the selected plants and their progenies is maintained. As a result, each progeny in every generation can be traced back to F2 plants from which it is originated. Such records are known as pedigree records. Pedigree selection, combining the desirable traits of two or more genotypes in a new improved variety or inbreed line. As we have seen previously, two plants are crossed and the third plant produced is found to be better than both the parents. The keys to successful pedigree selection. Detailed records should be maintained and most emphasis is given on selection of parents. Typically, one parent is already an improved locally adopted line. The other parent brings one or more desirable traits that are often well expressed. First one is selection of parents. The selection of parents is done on the basis of their performance over time and environments. Their adaptability should be more. Genetic diversity, geographical diversity should be present in plants. Their statistical performance should be good. Ability to pass desirable traits to progeny. They must have this capacity. Outline of pedigree method. In pedigree method, hybrids of F1 offspring with pedigree characters are selected. The seeds from each selected plants are collected and grown separately in rows to develop F2 generation. Then the best quality of F2 seeds are selected to develop F3. Similarly, F4 is developed and so on. The same process is repeated several times to obtain seeds of homozygous and uniform characters. This method is also known as pure line selection. An entire record is kept during whole process and this record is known as pedigree record. Here it is represented in this diagram. Two plants are selected. First one is parent 1 which is P1 and second one is parent 2 which is represented by P2. They are crossed to produce F1 generation. The seeds from F1 generation is used to plant F2. In F3 selection of superior rows are done and they are place planted in F4 generation. In F5 and F6 establish family progeny rows. Select individual plants to advance each generation. In F7 generation, conduct preliminary yield trials, select individual plants to advance. From generation F8 to F10, conduct advanced yield trials. And finally, it is released as a new cultivar. Third method is backcross breeding. In this method, F1 hybrids, which are produced by crossing of two different parents, is crossed with one of the parent. The main aim of the cross is to transfer a particular character of one parent to another in which this character is absent. Like we have taken the example of disease resistant, the similar we can perform with use of backcross breeding. The characters are disease resistance, drought resistance, etc. are introduced into crop plants. This is used for improvement of both cells and cross-pollinated plants. Suppose a superior character is absent in superior variety A. That means a plant A is having all the good character except it is prone to diseases. But this character is present in inferior variety B. Hence the desirable character of B is transferred to A by crossing A with B. This F1 hybrid are backcrossed with the recurrent parent A instead of cell pollination. The hybrids of first backcross are again crossed with recurrent parent A and this backcross is repeated several times to obtain offspring with desirable characters. Finally, the selected plants of backcross are self-pollinated to obtain homozygous and new characteristic population. Hybrids are chosen to improve the characteristics of the resulting plants, such as better yield, greater uniformity, improved color, and most importantly, disease resistance. An important factor is the heterosis or combining ability of the parent plants. The parent strains used are therefore carefully chosen so as to achieve the uniformity that comes from the uniformity of the parents. Because we have to pass the character of parents to the offspring, so parents must be chosen very carefully and the superior performance that comes from heterosis. Now hybridization in cross-pollinated crops. Production of hybrid. After identification of superior line of the hybrid seed is produced, there are three types of intervarietal hybrid. First one is single cross hybrid, three-way cross hybrid and double cross hybrid. Types of hybrid. Single cross hybrid, hybrid formed by cross between two varieties is known as single cross hybrid. Three-way cross hybrid, hybrid formed by a cross between single cross hybrid and an inbreed. Double cross hybrid, hybrid formed by a cross between two different single cross hybrids. Types of hybrid. In breed A, in breed B, this is seed parent and this is pollen parent. They are crossed, single cross hybrid is formed by combining the characters of two different parent A and B. Types of hybrids. Crosses between males and females can be made in 4 different ways to give rise to different kinds of hybrids. Single cross hybrid, crosses between two unrelated inbreed lines, that is one parent is A and another is B, both are crossed to get the hybrid plant. A field planted with single cross seed is impressive because plant height, ear height, pollen shedding and all the other characteristics are extremely uniform. Three-way cross hybrids. This cross is made between F1 hybrid of a single cross which is used as female parent and an inbreed line used as male parent. Three parents are involved in three-way cross hybrid formation. The female of three-way hybrid is a single cross hybrid. That means the plant which is produced in F1 generation by crossing two different parent A and B, while the male is an inbreed line C. Three-way hybrid seed is produced in single crosses plant so that yield and quality may be equal or nearly so to double cross hybrid seed. Advantages and disadvantages of three-way crosses are between those of double and single crosses. Three-way crosses are more variable than single crosses and less variable than double crosses. Double cross hybrids. Double cross hybrids are produced by crossing F1 hybrids of two separate single crosses. That means in first case, two parents are selected named as A and B. They are crossed to produce F1 generation. While in second case, two parents are selected named C and D and they are crossed to produce another F1 generation. These two F1 generation are crossed again to make double cross hybrids. Double cross hybrid seed production is a practical and economical way of producing educate seeds for farmers because both parents are hybrids having beneficial characters. They are more variable than single or three-way crosses, thus allowing breeders to bring more different desirable characters together in one single plant, which is not possible in single crosses. Here it is represented in the diagram. First we will see three-way cross hybrid. In this, two parents are selected, parent A and B. They are crossed to produce F1 generation and this F1 generation is again crossed with inbreed line. That means A and B produce this progeny and this progeny, this F1 progeny is again crossed by C. This type of crosses are known as three-way cross hybrid. While double cross hybrid. Double cross hybrid in first year two parents are selected. First one is A and second one is B. They are crossed and they form F1 generation. While another two parents are selected named C and D. They are crossed and produce another F1 generation. Now these two F1 generation in the second year they are crossed to produce double cross hybrids. Benefits of hybrid breeding. Higher yield is there. Hybrid vigor is there. Reduced plant stress, disease resistant quality can be developed, multiplication of superior varieties, availability of superior varieties to masses, maintenance of genetic purity, supply of high quality seeds, seeds have high germination status, seeds having high vigor and viability status, maintaining excellent health status of seed, securing high crop yields and income level to farmers. Naturally, when two good characters present in two different plants is bought in one single plant, the plant is going to be superior than its parents. Now, we are going to start another topic, somaclone and somaclonal variation. Soma means the somatic cells and clones means the generation. The term somaclonal variation was coined by Larkin and Screwcroft. According to them, somaclonal variation is the genetic variability which is regenerated during the procedure of tissue culture or plant variants derived from any form of cell or tissue culture. Genetic variation in plants that have been produced by plant tissue culture and can be detected as phenotypic traits. That means we can detect it from outside the plant body. Two types of characters are there, genotypic and phenotypic. Phenotypic is the external appearance. The genetic variability present among cultured cells Plants derived from such cells or progeny of such plant is called somaclonal variation or somaclones. The genetic variations are inherited by the clones of the treated plants. This kind of work is majorly done to achieve some of the beneficial properties of plants like disease resistance, increased pest resistance, stress resistance, nutritional quality improvement, etc. The recovery of genetic changes in plant generated from somatic cells thus offer an opportunity to unravel natural variability and to use this variation for development of new varieties for good purposes. Variation occurs in both qualitative and quantitative traits. Nomenclature of somaclones. The somaclones that are generated from tissue culture directly are regarded as R0 or R plant. The cell fertilized progeny of R0 is represented as R1 plant, R2, R3, R4, etc. These are subsequent generation, while other workers name it as SC1, somaclone 1, 2, 3 for their subsequent generations. Somaclones can be obtained by plant breeding, irradiation, genetic engineering, and tissue culture technique. Genetic variations in plants that have been produced by plant tissue culture and can be detected as genetic or phenotypic traits are known as somaclonal variation. Basic features. Variations for karyotype, isoenzyme characteristics, and morphology in somaclones may also observe. Calicones, that means the clones produced from callus, mericlone, clones produced by meristeme, and protoclone, clones of protoplast are produced. Generally, heritable mutations are found, which can persist in plant population even after their plantation into the field. Mechanism of somaclonal variation. First one is genetic. When changes are brought in their genetic constituent, those changes are heritable to subsequent generations. Pre-existing variation in the somatic cells of explant means the explant we have chosen for tissue culture already has some variation caused by mutations and other DNA changes occurs at high frequency. While second one is epigenetic, those changes which are not heritable, variation generated during the procedure of tissue culture caused by temporary phenotypic changes occurs at low frequency. Steps involved in induction and selection of somaclonal variation. Callus tissue, the process of organogenesis is done. By the process of organogenesis, by changing the concentration of the hormones, whole plant is regenerated. Then the plant is planted, hardening and selfing is done, and they are observed for phenotypic characters. Somaclonal variants are found. Causes of somaclonal variation, it may be physiological cause, genetic cause, or biochemical cause. Physiological causes. Exposure of culture to plant growth regulators. That means change in the concentration of required hormones. Different hormones affect the culture differently. Second one is culture conditions. Environmental condition as well as the conditions present in the culture room affect the cultural growth. Genetic cause. Change in chromosome number. It may be euploidy, that means changes chromosome sets, aneuploidy, changes in parts of chromosome sets, polyploidy, organism with more than 2 chromosome sets, and monoploidy, organism with 1 chromosome set. Changes in chromosome structure. It may be deletion, it may be inversion, it may be duplication, or it may be by translocation. Gene mutation. By transition, transversion, insertion, deletion, anything can be happened which generates somaclone variations. DNA sequence, change in DNA, detection of altered fragment size by using restriction enzymes. Third one is biochemical cause. Lack of photosynthetic ability due to alteration in carbon metabolism, biosynthesis of starch by carotenoid pathway, nitrogen metabolism, antibiotic resistance. Any could be the cause of somaclonal variation. Here in this diagram, this is shown. Leaf is taken as an explant, it is cultured, callus is formed, then by the alteration of hormone, organogenesis is induced, that means shoot regeneration, whole plantlet is formed. This plantlet is transferred to the field, screening for desired trait is done, and detection of somaclonal variation is done. Factors affecting variation. Selection of propagules. like cell, protoplast, chelae, selection of agent, toxin, herbicide, amino acid, technique used for selection, duration of cell culture, growth hormone effect, stability of resistant substance, in vivo testing procedure, ability for regeneration of plants, genotype and explant sources. Detection and isolation of somaclonal variants. First one is analysis of morphological character. Qualitative characters like plant height, maturity date, flowering date, size of leaf. Quantitative characters like yield of flowers, seeds, and wax content in different plant parts. Second one is variant detection by cytological studies, staining of meristematic tissue like root tip, leaf tip, which provide the number and morphology of chromosome. That means first we have to identify the morphological characters and then we will go for cytological studies. Third one is variant detection by gel electrophoresis. Change in concentration of enzymes, proteins, and chemical products produced by plants like pigments, alkaloids, amino acids. It can be detected by their electrophoric pattern. Fourth one is detection of disease resistant variant. Pathogen or toxin responsible for disease resistant can be used as selection agent during culture. Plants are grown in this toxin and only resistant plants can be found alive. Other plants will die. Variant detection by DNA contents. Cytophotometer detection of fluorescent stain nuclei can be used to measure the DNA content. Isolation of somaclonal variants. A number of selection systems are now being used to select the variants. First one is without in vitro technique. In this, the whole plant is regenerated, transferred to field, and then the characteristics are observed to analyze somaclonal variants. In this technique, unorganized callus and cells grown in culture for various periods on a medium that contains no selective agents are induced to differentiate in whole plant. Without the selective agent, the callus is regenerated into a complete new plant. An explant, leaf, stem, root, etc. is cultured on a suitable medium, supplemented with growth regulators. The unorganized callus and cells do not contain any selective agent at any stage of their growth. That means toxic or inhibitory substances are not present in the medium. These cultures are normally subcultured and transferred to shoot induction media for regeneration of plants. The so produced plants are grown in pots, transferred to fields and analyzed for somaclonal variants. Second is B. With in vitro technique, cell lines are screened for cultures by their ability to survive in the presence of substance in the medium that may be toxic or under condition of environmental stress. That means cell lines are introduced to environmental stresses or toxic substances in the medium. The differentiated callus obtained from an explant is exposed in the medium to inhibitors like toxin, antibiotics, amino acids, etc. Selection cycles are carried out to isolate the tolerant callus cultures and these calli are regenerated into plants. That means those callus are selected which can tolerate this stress and regenerated into new plant. The plants so obtained are in vitro screened against the toxin or pathogen or any inhibitor we have chosen. The plants resistant to the toxin are selected and grown further by vegetative propagation or by self-pollination process. The subsequent generations are analyzed for disease-resistant plants against the specific pathogen. Now here it is represented in the diagram. One plant is selected as a mother plant or a light plant. Explant is taken out. Now explant is put in a medium to form a callus. This callus is proliferated and maintenance of callus is done. Now the small piece of this calli is introduced in the medium which is already having toxins. In an alternate method, pathogenic organism is cultured. Purify the cultural filtrate. Isolation of toxin is done. Toxicity determination for lethal concentration is checked out. Then selection trials or selection cycles are done. Isolation of tolerant calli. The calli which is tolerant to this toxin is taken out, regenerated to form the whole plantlet. In vivo screening against toxin and pathogen is done. Then by the process of self-pollination or by vegetative propagation, progeny clones from each plant is formed. Test for disease resistance is done. Generation of disease resistant plant. Now these plants are subjected to agronomic trials. Advantages of somaclonal variation. It helps in crop improvement, creation of additional genetic variations, disease and stress resistance is introduced in plants, increased and improved production of secondary metabolites, selection of plant resistance to various toxins, herbicides, high salt concentration and mineral toxicity, suitable for breeding of tree species, mutants for agronomic characters. Limitations. Sometimes it leads to undesirable results. Selected variants are random and genetically unstable. It requires extensive and extended field trials. Not suitable for complex agronomic traits like yield, quality, etc. Thank you.